Electron spin resonance or ESR spectroscopy. Origin and properties of ESR spectra. An electron also possesses a spin magnetic moment that can assume two orientations in space and thus should interact with magnetic field in the way just described. For nuclei in NMR spectroscopy. In most chemical compounds, electrons are paired, so the net magnetic moment is zero, and there is no condition of resonance for absorbance of radiation. However, there do exist substances that possess an unpaired electron. For example, free radicals, certain ions of transition metals, excited photochemical intermediates in triplet state, and molecules such as oxygen that have a triplet ground state. Such substances are called paramagnetic, and many can be detected by electron spin resonance spectroscopy. This technique is also known as electron paramagnetic resonance or EPR spectroscopy. The electron spin magnetic moment is about 10 to the power 3 times greater than a typical nuclear magnetic moment, so that energy of interaction with an applied field is much greater in ESR than NMR. This means that for a particular magnetic field strength, the frequency of radiation absorbed is much higher in ESR than in NMR, and in fact in microwave region rather than radio frequency region encountered in NMR. This difference has two important consequences. First, ESR is much more sensitive than NMR and can be used with substances in solution at a concentration of 10 to the power minus 6 molar rather than higher concentrations of 10 to the power minus 3 molar needed for NMR. The second, the technology for detecting electron spin resonance differs and yields a spectrum that is not an absorption spectrum but the first derivative of the absorption spectrum. Now, what is this first derivative of absorption spectrum? You can see here this is the absorption spectrum as seen in NMR, and first derivative of absorption spectrum looks like this, and real ESR line comes like this. That is, we get sharp peaks in this spectrum and this type of graph, that is it is going upwards and downwards both, while in only in absorption spectrum, as we see in NMR, and this curve goes upwards only. Now its explanation. What is meant by that is shown in figure. Instead of symmetric peak, instead of wait, Instead of symmetric peak, a peak trough configuration is observed. This peak trough pair is nonetheless called a line. It might be thought that the peak trough configuration would have disastrous effect on the resolution of a spectra, as would be in the case of NMR spectrum if every peak becomes a peak trough. However, this is not the case because a typical ESR spectrum does not contain tens or hundreds of lines, but usually fewer than 10 lines, which are not closely spaced. This is because there is rarely more than one unpaired electron in the compound. So NMR में बहुत सी लाइंस मिलती हैं as compared to ESR. इलेक्ट्रॉन में ESR में इलेक्ट्रॉन अनपेयर्ड इलेक्ट्रॉन मुश्किल से एक होता है, while NMR में बहुत ढेर सारी एब्जॉर्प्शन पीक्स मिल सकती हैं. सो यहाँ पे कोई फर्क नहीं पड़ता अगर हम एब्जॉर्प्शन स्पेक्ट्रम का फर्स्ट डेरिवेटिव देखते हैं. द फ्रीक्वेंसी ऑफ वेट The frequency of ESR lines is determined by the energy difference between two spin states of electrons when in a magnetic field H. This difference delta E is given by is equal to G EH by 4 pi m H, in which E and M are the charge and mass of the electron, H is Planck's constant, G is a universal constant called G factor, with a value of 2.00232 for a free electron. The expression EH upon 4 pi m is called Bohr's magneton and is usually denoted by beta. The frequency nu of the radiation corresponding to the transition is defined by delta E, which is equal to h nu. So that this frequency is equal to G beta h by capital H by Planck's constant. In ESR, just as in NMR spectroscopy, it is not the strength of the applied field, but the strength of the field seen by unpaired electron that determines the resonance condition, that is the frequency. This change in frequency from that of a free electron occurs in two parts. First, there is a local magnetic field produced by a molecule that contains unpaired electron. Second, there are fields produced by other nearby molecules. The first effect is larger one and usually expressed as change in the value of G factor, that is an effective G factor. The measurement of G for unknown line is a valuable aid in identifying the source of light. This is of course equivalent to contains the unpaired electron. Second, there are fields produced by, this is a repeat, so I should strike this. This is typing error, so This is of course equivalent to identification by the value of chemical shift in NMR. The second effect provides the information about molecules that is desired. How this is done is described later. There is another consequence of local magnetic field on energy of the unpaired electron. The magnetic moment of the electron also interacts with magnetic fields of nearby spinning nuclei, usually the nucleus of the atom carrying the electron. This interaction gives rise to splitting so that a cluster of lines rather than a single line results. This is known as hyperfine structure. The number of lines per cluster is determined by the number of possible orientations of magnetic moment of the particular nucleus with which the electron interacts. In general, if the spin quantum number of the nucleus is I, the ESR spectrum has 2I plus 1 lines of equal intensity. Thus, if an unpaired electron approaches a N14 atom whose nucleus has a spin of 1, there will be 2, 1 plus 1, that is 3 lines in the cluster. This is shown in the spectrum of free radical ion SO3 whole twice NO minus. We will see it just now. If the electron is near proton for which I is half, there will be 2, 1 upon half, 1 upon 2 plus 1, that is 2 lines. The spacing between adjacent lines of a cluster is constant for a particular cluster. But that differs for each molecular type. The magnitude of the spacing depends on several factors that are beyond the scope of our study. An electron may interact with several nuclei since the spacing produced by interaction with each nucleus will differ. The total number of lines is a product of number produced by each nuclear interaction. Thus, if an unpaired electron is very near both an N14 atom giving 3 lines per cluster and a proton giving 2 lines per cluster, and if the proton induced spacing is less than N14 induced spacing, the spectrum will consist of 3 pairs of 6 lines as shown in figure 1722. So let us see figure first. This was first figure 1721 in which ESR spectrum of radical ion is shown. The free electron is on N atom and thus interacts with the N nucleus. The nuclear spin of N14 is 1, so that hyperfine splitting to yield 3 lines occurs. Now let us see the 1722. This figure shows an ESR spectrum showing splitting caused by 2 different nuclei. The distance A and B are separations induced by proton and N14 nucleus respectively. This is because of proton near electron, unpaired electron, and this is the distance between nucleus of the same atom. Often an electron interacts with several protons that are magnetically equivalent. An example of this interaction is interaction between the free electron of the carbon of ethyl, sorry, an example of this example is the interaction between free electron of carbon of ethyl radical CH3, CH2. So free electron of this ethyl radical and two methylene protons on the same atom. Each proton produces a pair of lines, but the higher frequency line of one pair has the same interaction energy as the lower frequency line of the other pair, a phenomena already seen in case of NMR as shown in figure 17.6 and 17.7. That is this I'm telling you ESR from the same book where these figures were shown for NMR spectrum. So as about ESR spectra, three lines are seen and central line has twice the intensity of each of the outer lines. In general, if there are n equivalent protons, the spectrum consists of n plus 1 line whose intensity distribution is given by coefficients of the binomial distribution, namely intensity distribution. This is the distribution of coefficients. Intensity distribution is shown like this. This is shown in 7-line spectrum of benzene radical anion. 7 lines was shown in figure 1723. So let us go to 1723 first. Wait. This is 1723. The ESR spectrum of benzene radical anion in solution. You can see this intensity of middle line is highest as we studied just now. This spectrum demonstrates one kind of information obtainable from ESR spectroscopy. If the free electron were localized on one of the 6 carbon atoms of this radical anion, the 6 protons will not be equivalent. Instead, the interaction would be with single proton on that carbon atom and there would be 2 lines. There is no interaction with 2 adjacent carbon atoms because the nuclear spin of carbon is 0. Carbon 12 is 0. However, the 7 lines indicate that the free electron interacts with each of 6 protons. Hence, the free electron cannot be localized on a particular carbon atom but must move through the benzene ring, interacting equally with each proton. Uses of ESR spectroscopy. Existence of an ESR spectrum and the pattern of the splitting can both give information in biochemical analysis, and few examples are given here. First is elucidation of the mechanism of an enzymatic reaction. Site of binding of copper ion by a protein, binding of calcium ion to a bacterial enzyme, fluidity of the interior of erythrocyte membrane can be. We can get a spectrum from there so that we will deduce how ordered various molecules are there in the membrane. The dynamic state of the membrane can be found out. फ्लिप फ्लॉप मैकेनिज्म के लिए भी इसको यूज किया जा सकता है. The depth of crevice surrounding the binding site of a protein. आपको पता है कि एंजाइम हो या प्रोटीन हो, बहुत से ग्रूव्स पाए जाते हैं ग्लोबुलर प्रोटीन्स की सरफेसेस पे, तो उनकी डेप्थ पता की जा सकती है. So let us see these methods again. We need to remember some of these examples. Like elucidation of the mechanism of enzyme reaction. You can copy it down. The reaction ABC. AB gives C plus D as shown here. Enzyme plus A, enzyme A, enzyme A plus B, enzyme AB, then enzyme AB gives rise to AB plus enzyme, and AB splits into C plus D. Second option is enzyme plus A, enzyme A goes to 2C, C plus B, then C plus B, and finally B dash plus C dash, and they make D. There are no free radical intermediates in scheme 1, but there are two such intermediates in scheme 2. If lines observed in an ESR spectrum of reaction mixture according to scheme 1 is unlikely, from the change in the spectrum with time, it may be possible to determine whether B free radical or C free radical are intermediates, because these carry free radicals. In scheme 2, there are two different compounds containing free radical, thus there should be two sets of lines which may overlap. Furthermore, from the proposed structure of B and C, the splitting pattern of each set can be predicted. If a spectrum does not support the presence of B and C, another scheme having free radical intermediate would have to be proposed. In no case would a radical-free mechanism be possible. तो फ्री रेडिकल तो होना ही होना चाहिए, फ्री इलेक्ट्रॉन होना चाहिए जो कि स्पिन कर रहा हो, तब इस स्पेक्ट्रोस्कोपी को यूज किया जा सकता है. In no case would a radical free mechanism be possible. If, on the contrary, no ESR spectral lines are seen, one may not conclude that the mechanism does not include a free radical intermediates, because the lifetime of intermediate may be so short that its concentration is too low to be detectable. Observation of this sort have been extremely valuable in understanding the mechanism of molecular damage by ionizing radiation such as X-rays, because well-known radicals such as OH have been detected when water is irradiated. Now, next example is site of binding of copper ion by a protein. An enzyme binds copper ion, but the binding site is unknown. It has been suggested that ion binds to an imidazole N of histidine. This would produce a splitting into 3 lines because the spin of N14 is 1. Other known sites of binding of copper ion are oxygen and sulfur, oxygen 16 and sulfur 52. Both have 0 spin and there would be no splitting. 3 lines are observed which suggest that binding to nitrogen is possible. An ESR spectrum was also obtained from N15 substituted protein. This isotope has a spin of half. Confirming first conclusion, only 2 lines are observed. Thus, the copper ion is bound to nitrogen atom, although there is no proof from this experiment that it is histidine nitrogen. Binding of calcium ion to bacterial enzyme can also be found out. Likewise, I'm going through it very slowly so that you can read it. I'm not going to tell each and every example in detail, but this is the text. You may read and note down. You read and write this example also. Now there is a next example, the fluidity of the interior of erythrocyte membrane. When red blood cells are placed in a buffer of low osmotic strength, the cells swell and become porous. Hemoglobin and other cell contents leak out, leaving a pure cell membrane. The fatty acid, stearic acid, is easily incorporated into this cell membrane, and its carboxyl group may be located at either the inner or outer surface of the membrane, while the organic chains remain inside. 2 stearates containing a nitroxide have been synthesized with spin label either near the carboxyl group or at the other end. These molecules have been separately incorporated in 2 membrane preparations and ESR spectra of each were obtained. As shown in the figure, the spectrum obtained when structure 1 is present is much broader than seen when the structure 2 is present. By comparison to standards, we can see this. If this type of graph is coming, then membrane is very rigid. We know that membranes are fluid, but this type of frozen, under frozen conditions, membranes are rigid, less mobility of uh, that fluid is seen. When temperature is reduced, moderately immobilized, little tumbling is seen, and increasing the glycerol concentration. Tumbling is further reduced, freely tumbling, there is no glycerol, and at room temperature, this type of curve will be obtained. And this gives the idea about the fluidity of membrane components. You can see what is written here. Typical ESR spectra showing line broadening as the mobility of a spin label is reduced. जो हमने जिस चीज से हमने लेबल किया है मेम्ब्रेन को, अगर वो सिग्नल उसकी मोबिलिटी कम होती है तो स्पेक्ट्रा ब्रॉड आता है. Narrowest line in aqueous solution and at room temperature is found. ये इस तरह का स्पेक्ट्रा आएगा अगर रूम टेम्परेचर पे लिया जाए. Aqueous solution है. Then they broaden if glycerol is added to increase the viscosity. Again viscosity बढ़ेगी तो fluidity घटेगी. Broadening continues as the temperature is lower. Again temperature नहीं है, temperature कम है, kinetic energy कम है, तो membrane के different components move नहीं करेंगे. When the sample is frozen, the line at the far right is so broad that it is difficult to discern. Freeze कर दिया तो बिल्कुल ही movement नहीं हो रहा है. Okay. Data suggests that the carboxyl end of stearic acid is not free to move and organic end moves freely. That is, the interior of membrane is sufficiently fluid to allow substantial rotational freedom of incorporated fatty acid molecules. Note that a rigid type spectrum can be seen even though membranes themselves are tumbling in solution. This is because membranes move only very slightly during very small time interval required to absorb high frequency microwave radiation. Studies of this sort have been used to understand membrane function. For instance, addition of other substances can increase or decrease fluidity, at the same time affect the rate of transport of various molecules. Furthermore, by lowering the temperature, it is possible to determine the temperature at which internal region of membrane becomes solid-like. This has correlated with transmembrane transport rates. You can see ESR spectra for two spin-labeled derivatives of stearic acid incorporated into erythrocyte membrane. Spectrum 2 is narrower when the spectrum 1 narrower than spectrum 1, so that type 1 molecules are more mobile than type 2 molecules. So जो fatty acids होते हैं उनका पोलर हेड पे एक तरह का लेबल चिपका दिया, दूसरे पे दूसरी तरीके का, so that local we can get uh, the idea about the local environment within the fluid membrane under study. Similarly, dynamic state of the membrane can be studied. वो उसमें मॉलिक्यूल्स आपस में कैसे इंटरेक्ट कर रहे हैं, कैसे हिल रहे हैं, कितने समय पे क्या हो जाता है, ये सारा इसमें से पता किया जा सकता है लेबल्स यूज करके, जिनको इंसर्ट करने का टाइम देंगे वो लेबल अंदर हो जाएंगे. अगर मेम्ब्रेन के एक लेयर में से this fatty acid comes downwards layer or this goes upwards, then that phenomena is known as flip flop. And we can find out these changes because if a spin label is there, then that informs the give idea about the microenvironment surrounding these molecules. Besides, depth of crevice surrounding the binding site may be found out by the use of this spectroscopy. So these were the various applications, principle and applications of ESR spectroscopy, which is also known as electron paramagnetic resonance (EPR). Now about instrumentation. This is a schematic diagram of electron spin resonance spectrometer. Okay, these two are magnetic poles. Sample is kept here. Electron oscillator produces microwave radiation, which comes here, and reflected microwave radiation is detected by crystal detector, CRO, and sweep supply, and here you will get the spectrum. This wait. The basic components of an electron spin resonance spectrometer are shown in the figure. Monochromatic microwave radiation is produced by Electron oscillator, the wavelength being of the order of 3 into 10 to the power minus 2 meters, 9000 megahertz. Sample must be in solid state, therefore bilateral samples are usually frozen in liquid nitrogen. The microwave radiations are passed towards the sample and reflected microwave radiation is detected by a crystal detector which converts the microwave into electrical current. This is amplified by amplifier and the signal from the amplifier is recorded by a cathode ray oscillograph. Applications we have already seen, but in short, it is used as investigative tool for the study of radicals formed in solid materials. Since the radical typically produce an unpaired spin on the molecule from which an electron is removed, study of radicals produced by the ionizing radiation gives information about the location and mechanism of radiation damage. ESR is used to detect the free radicals. Free radicals are found in metabolic pathways and as degradation products of drugs and toxins. In general, free radicals are found in low concentration in tumors than normal tissues. ESR is used to detect, sorry, electron spin resonance is one of the main methods used to study metalloproteins, particularly those containing xanthine oxidase, which contains molybdenum, copper, cytochrome oxidase, and copper blue enzyme, and iron, which is present in cytochrome and ferridoxin, etc. Both copper and non-heme iron, which do not absorb radiation and ultraviolet or visible regions, possess ESR absorption peaks in one of their oxidation states. Hence, the appearance and disappearance of their electron spin resonance signals are used to monitor their activity in multi-enzyme systems of intact mitochondria, chloroplasts, as well as in isolated enzymes. In metalloproteins, the metal ion has a characteristic number of ligands coordinated to it in a definite geometrical arrangement. These ligands are frequently amino acid residues of the protein. ESR studies reveal the distortion in structural geometry, and the distortion may be related to biological function, intramolecular motions, and lateral diffusion of lipids through the membrane may be observed and measured. This study is achieved by either concentrating the spin labeled lipid into one region of the bilayer or randomly incorporated, usually into model membranes. So this is all for ESR.